Chapter 13 looks at the diversity of microorganisms. Microbes are incredibly diverse, and they range a lot in their habitats and their size. For example, if we look at this scale over here, viruses, and we'll talk more about each of these as we go through the chapter, viruses are very, very small, and microorganisms range from about the size of viruses all the way up to the size of amoeba, which is about 1 millimeter. So if you look at a ruler on the metric side, each of those little small lines are a millimeter. So they range vastly in size. To give you a good visual example of these sizes, if a virus is the size of the head of a pin, then the equivalent size amoeba would be the size of an oak tree. So this is an incredible difference in size for microorganisms. They're also incredibly diverse, and we'll talk more about them as we go through, but they basically can inhabit any type of environment anywhere on the planet that we know of. Even on your own body, there are trillions and trillions of microbial cells. Just a couple of examples. Living in your mouth, you have 500 different species of microorganisms on average that live in your mouth. Now, each of those 500 species has millions of individual representatives of those species. So you have millions and millions of bacteria living in your mouth. Your intestinal tract only has about 400 species, so there are actually fewer microbes living in your digestive system than in your mouth. And your skin has only about 200 species on average living on your skin. You combine all of those and look at the total number of microbes living in your body and also on the surface of your body, and they outnumber you 10 to 1. So if you look at the number of human cells in your body compared to the number of microbial cells that live in and on you, there are 10 times more microbes than human cells. So you're a minority within your own body. There are different types of microbes, and we'll look at each of these as we go through the chapter. First, we'll look at bacteria. And bacteria we've talked about before. We've actually worked with them in lab. They're single-celled microorganisms, and they're fairly simple in their body structure. Archaea looks very similar to bacteria. So again, a very simple body structure, single-celled. However, looking at their chemical composition and their genetics, they're incredibly different from bacteria. In fact, they're closer to eukaryotes, which is the group humans are in. So technically, archaea, even though they look just like bacteria, would be more closely related to humans than they would be to other bacteria. We'll also look at protists, and protists are a type of eukaryotic cell, so now they have a nucleus and other organelles. And kind of off to the side here are viruses, and we'll also look at viruses in this chapter. And viruses are interesting because they're not truly living organisms. They're technically not alive because they are missing some of the characteristics that humans use to define life. So a virus has no metabolism, and a virus can't reproduce on its own, but it does have genetic material. So we'll look at viruses more later in this chapter and why some people describe them as non-living organisms. Others would argue with that and say, well, they are living organisms, and there's a debate there. So viruses really are on that kind of gray area between a living organism and a non-living. They're in between. We'll start with bacteria and then move on to the other types of organisms. So bacteria we've looked at before, and we know their basic parts. They have one main chromosome internally, and they also have smaller piece of DNA called plasmids. And in fact, we worked with those plasmids when we did our pea-glow experiment. We moved a plasmid containing DNA from a jellyfish into an E. coli bacteria to make them fluoresce or glow in our UV light. They also have a phospholipid membrane, and that's surrounded by a cell wall. Now that cell wall helps hold their shape, and there are a variety of shapes. So there's round shape or spherical bacteria, there's rod shaped bacteria, And there's spiral-shaped bacteria. Those are the three most common types of bacteria, but there are lots of other shapes out there as well. But these are the three most common, the ones you're most likely to encounter if you took like a microbiology class, and these would be the most common organisms living on your body. Another thing about that cell wall, in addition to holding the shape of the bacteria, that cell wall can be composed of different layers and different, structures within those layers, different thicknesses of those structures, and you can detect that using something called a Gram stain. And if you've ever taken a microbiology course or take one in the future, you'll become very familiar with Gram stains. And it's a process where you take bacteria on a slide and you run it through several different steps. At the end of that, you should have bacteria that are stained purple, and those purple ones are going to be Gram positive. And then you'll have ones that are lighter in color or a pinkish color, and those are Gram negative. Now, this is a very important test to know because the positive and negative colors, the purple or pink, is determined by the structure of the peptidoglycan on the cell wall of these bacteria. And that's really important because certain antibiotics like penicillin do not work against Gram-negative bacteria. Just based on that structure, penicillin doesn't work against them. So it's important to know what type of cell wall structure you have in your bacteria, whether it's Gram-positive or Gram-negative, so you know what antibiotics to treat the bacteria with. Bacteria also look very different on plates. If you plate them out and look at them, there's different colony sizes, structures, different colors, different growth medias that they'll grow on. And you'll be able to see some of this when we do our environmental sampling lab and also just look at other microbes in our microbiology lab. More about bacteria. Their generation times are very short, so under ideal conditions, something like E. coli can divide every 20 minutes. So that's a generation every 20 minutes. And so even though mutations or mistakes in copying the DNA are rare, if you copy the DNA every 20 minutes, you're going to be able to rapidly change and evolve your DNA, which is a problem with antibiotic resistance. One of the big problems that we have is bacteria can evolve so quickly to develop new biochemical pathways to resist our antibiotics. We'll talk more about that later in the chapter. But just as a reminder, we've looked at this before in the cell division chapter, but for bacteria to divide, all they do is copy their DNA. So here they've copied their main chromosome, and they also copy any plasmids that they have. It stretches just a little bit, and then the cell divides in two so that the resulting daughter cells now are exactly the same as the parent. They have the same chromosomal DNA and plasmids with some possible mutations in there. And that's a type of asexual reproduction called binary fission. Even though bacteria use asexual reproduction, they still can get DNA from other bacteria, which we normally think of in sexual reproduction, where you mix DNA from different organisms to increase the genetic diversity. Well, not only do bacteria have the potential for mutating very rapidly because of their short generation time, they can also share DNA between adults, something called horizontal gene transfer. We're very familiar with vertical gene transfer, going from parent to offspring to offspring to offspring, and that's vertical. It goes directly in a line. However, horizontal gene transfer, they can transfer to any other bacteria in the adult stage. And it doesn't have to be the same species. If one bacteria recognizes the other as a bacterium, they can do something called conjugation, where the initiator of conjugation can basically lasso and grab a recipient, pull it close. It forms a conjugation tube or an open tunnel between the two cytoplasms, and it will pass plasmids across. Not the main chromosome, usually, but there are ways to get the main chromosome across too, but it will pass plasmids across. And that's kind of scary because it's those plasmids that generally contain information on how to resist antibiotics. So it can pass it directly to another bacterium. Also, transduction is another way to transfer DNA from one bacteria to another, and it uses a virus intermediate. So there are viruses that infect bacteria, and if a virus infects a bacterium, when it assembles new viral particles, if it makes a mistake and puts bacterial DNA in the new virus instead of viral DNA, that virus will go out and infect another bacteria and inject bacterial DNA instead of viral DNA because of the packaging mistake. So now we've transferred bacterial genes from one bacteria to another just using a virus intermediate. Some bacteria can also absorb DNA from their environment in a process called transformation. And some can do this naturally in kind of conditions. However, we can also force bacteria to do this in the lab. This is what we did with our E. coli cells when we inserted our pGlow gene. We forced them to absorb that DNA from the environment. So these are 3 different ways that DNA can transfer between bacterial cells, and so how they can mix up their genetic information. If one single bacteria develops resistance, they're going to be able to pass that gene along in one way or another. Bacteria are also incredibly diverse metabolically, and they can metabolize pretty much anything. Even kind of as time goes on, we're finding more and more man-made substances that bacteria learn how to digest and metabolize and use as a food source. And a couple of terms that I'll want you to be familiar with on how we refer to what bacteria eat. Let me break this word down a little bit. This is called chemoorganotrophs, and troph means to eat something. That's eating. Organo just refers to organic, and the chemo part just refers to the fact that it's using kind of a chemical or a molecule structure. So basically, this chemoorganotrophs means that the molecules that they eat are organic. And so they eat organic molecules. Again, if we look at chemolithotrophs, we know troph means to eat, and we know chemo means the molecules that food is composed of. Litho is rock. So these literally are rock feeders. So they get their molecules from rocks or other inorganic, non-living things. So ammonia, hydrogen sulfide, hydrogens, all of those things can act as food sources for a chemolithotroph. And these are sometimes problematic. This is what happens at mines when they have kind of their waste product. It will interact with these chemolithotrophs, and a byproduct of their metabolism is they create acids, very strong acids, and that gets into the water and kills fish. This is why mines have to be careful not to contaminate waterways, because interaction with the bacteria will harm the environment. Another type of bacteria that's not a chemo using molecules but a photo, meaning it uses light. This is a photoautotroph, and again we see troph, so that is a feeder or eating something. Photo means light, and auto means they can do it on their own. So these feed themselves light is what that term says. So these eat light. They can use photosynthesis, capture energy from the sun to drive their metabolism. And it's these photoautotrophs, cyanobacteria, that were some of the first living organisms to emerge that started generating oxygen as a waste product. And that's what caused our Earth's environment to change from a very toxic mix of methane gas that we spoke of in the previous chapter to what we have currently, which is about 21% of the atmosphere is composed of oxygen. That allows us to breathe and other things to survive and breathe as well. And even now, today, I know we usually think of trees and plants as producing oxygen, but they still produce a vast majority of the oxygen. Globally, the first about 2-3 meters of ocean water is very full of these photoautotroph cyanobacteria that are still busy producing oxygen, and they still make about 40% at least of the oxygen that we're breathing today. In addition to generating oxygen, there are lots of other beneficial bacteria You've heard of eating yogurt because it has live cultures. It's good for digestion. One of those live cultures is Lactobacillus. This bacteria is good for your digestive system and very beneficial, especially if you're on antibiotics that would kill off harmful as well as helpful bacteria. It's important to replace those live cultures, so eating yogurt after you've been on antibiotics is beneficial. Also, lots of products that we make, so cheese, beer, wine, and vinegar, all of those are made possible because bacteria ferments the sugars in those for things like wine and beer, and also cheeses and vinegar, and turns it into a different product. So it curdles the cheese and produces the wine, beer, and vinegar as a waste product. And so that's important. Also, if you have a normal flora or the proper types of bacteria living in your digestive tract, you're unlikely to get sick. However, if you don't have those normal organisms there, then that opens living space or opportunities for diseases to come in and make you ill. And there are actually even therapies where they take these living organisms from other people's digestive tracts and inject that into people who are sick and have disease problems to replace their living cultures. And that's a type of probiotic therapy. So probiotic therapies use living microorganisms to treat illnesses. And there's lots of probiotic therapies out there. So digestive tract problems, so severe diarrhea, you could treat that by taking a healthy person, removing some of their digestive tract bacteria and transplanting that into the person who has the severe illness. Can also be used to treat bladder infections. Lots of different things are now using probiotic therapy. In addition to being helpful, there are also lots of bacterial diseases. So pathogenic bacteria are disease-causing. So pathogenic means that it causes disease. There are some bacteria that are always pathogenic. So if you're exposed to them, they're always going to make you sick. And those would be cholera, plague, tuberculosis. Those will always cause disease. However, some bacteria depends on their environmental conditions. So acne, most people are probably carrying the bacteria that causes acne. However, it's changes in hormones and stress levels that allow those bacteria to create pimples. So even though you may be carrying acne bacteria, you have clear skin. Also strep throat. So some people can carry the bacteria for strep throat but not actually be sick. Scarlet fever and flesh-eating bacteria are the same. Many people carry them. In fact, you might be hosting them right now, but it doesn't make you sick. However, somebody else with a slightly different immune system could become sick with those. One of the first cases where a disease was stopped by preventing people from coming in contact with bacteria was in London. Dr. John Snow started mapping cases of cholera that were found, and he discovered that most of them clustered around one area. And this is back before we had indoor plumbing, and people would go out to common community wells or pumps and get their water and carry it back home. And they found that this one pump had the most cases surrounding it, and the other cases further away were people who had visited that area or used that pump. So he got permission from the city to remove the handle. All he did was take the handle off the pump, and the outbreak stopped. The water underneath that pump had become contaminated with fecal matter containing cholera, and that had caused anybody exposed to it to become sick. And so that was one of the first cases where they stopped a bacteria from traveling through a population. Another thing about bacteria that I've mentioned earlier is it can become very drug resistant or resistant to antibiotics, which is frightening considering that the first antibiotic, penicillin, was developed kind of midway through World War II. So we've just started using antibiotics during World War II, and now almost all of them have some type of resistance to one, if not all, medicines that we know of. An example, S. aureus, is a very, very, very common bacteria on the skin. In fact, most of you are probably carrying it right now. However, some strains of it tend to be more aggressive than others and can start causing problems. And there's one called MRSA, and this is resistant, so I think it's methicillin-resistant Staphylococcus aureus. This is resistant to even our most powerful antibiotics that we know of to treat this infection. And so this has become very, very lethal. And in fact, in the U.S., currently, there are more deaths annually from MRSA than there are from HIV. So this is an incredible problem to people's health, and this is very common in hospitals. Some different ways that MRSA and other bacteria are able to fight off antibiotics are, again, DNA on those plasmids will carry information for new antibiotic pumps. So once the bacteria is exposed to antibiotics and those antibiotics get into the bacterial body, it just grabs them and pushes them back out. So it keeps them outside the cell and they're not destroyed by the bacteria. They've also created binding proteins. So these are proteins or molecules that as soon as the antibiotic is added, these proteins will attach to the antibiotic, preventing it from working. And also enzymes that will just chew up and destroy the antibiotic. So these are just a couple different ways that bacteria have become resistant using pumps, binding proteins, or enzymes. One of the problems with resistant bacteria is it's being caused by humans. So if you've ever had a prescription from your doctor, you go to the doctor, you get a prescription for a bacterial infection, do you always, absolutely every time, take it as directed? If you answered no or yeah, mostly to that question, then you're actually helping to contribute to resistant bacteria. If you take it as directed, it will prevent resistant strains from developing. However, if you don't take it as directed, what happens is you are exposed to the antibiotic and it will start killing off any bacteria that are susceptible. And so only the more resistant ones will survive. And if you continue taking the antibiotic, you should be able to get rid of them completely. However, if you stop taking the antibiotic partway through, then you're still going to have some of those resistant, harmful, the harmful ones are the green ones here, those harmful bacteria around. And those harmful bacteria now are more resistant to your medication than the ones at the beginning, because those are the survivors, the ones that could survive just a little bit longer. And so now these will grow and multiply and spread that resistance gene. And if you go back to the doctor and say, oh, the medication you gave me was awful, it didn't help because you stopped taking it partway through or didn't take it exactly on time as directed, then they'll give you another antibiotic and start the cycle all over again. This is why there are some bacteria that are resistant to everything that we have. It's a lethal infection. If you get infected by certain types of bacteria, there's nothing doctors can do, even with all of our modern medications. And it's not just human prescriptions, it's also a huge problem with agriculture. So there are 25 million pounds of antibiotics produced annually that only go to agriculture. That's 8 times more antibiotics than all of human medicine. And what happens in agriculture is animals are grown in very crowded conditions. They're fed diets that they're not normally, naturally supposed to eat. So cattle that are fed corn, they actually develop digestive tract problems and can get infections and not put on weight. And so as a preventative measure, farmers start adding antibiotics to the feed. So they feed them an unhealthy diet. Cattle are designed to eat grass, not corn, but it's much easier and faster to grow them on a corn diet. So we start adding antibiotics to the corn diet so that way we'd be treating any digestive tract problems as they eat. However, problem with that these low doses of antibiotics constantly being given, those bacteria have basically evolved and are no longer susceptible to those antibiotics, and so they have to switch antibiotics and do something else. Because of that constant low-grade exposure, we have brand new versions of bacteria cropping up in agriculture all the time that are resistant. And that's a problem for the food industry, but also some of those can then get transmitted to humans. So now we have a new human bacteria that's resistant to this variety of antibiotics that have been used in the cattle feed, or the pig feed, or the chicken feed, or in the aquatic tanks used to grow our fish. So all of those environments heavily coated with antibiotics, encouraging resistant strains and causing a problem because those strains either can get into humans directly or through things like conjugation and transduction, they can inherit or pick up those genes along the way. So a human bacteria can pick up resistance genes from a cattle or a pig or a chicken virus. So now that human bacteria is resistant to the antibiotics. And read the article, this is one of the assignments, the Big Pig Problem article, and that will talk about how some farmers are now trying to get off of using antibiotics and kind of their success with that. Something else that we've already, or we're going to talk about in lab, are STDs. And in lab we'll talk about bacteria and viruses, so we'll cover those in lab, and also we'll look at lice in lab. A couple of things that I don't normally mention in lab are there are protists and fungus that can also be STDs. So protists like trichomonas, these are small swimming living organisms, and again they're eukaryotic, and they can also be passed through sexual contact and have very uncomfortable symptoms. And yeast infections can be caused by taking antibiotics that kill off your normal bacteria, and so the yeast is then able to grow out of control, or you can pick up yeast from other individuals. So that is also an STD.